anyway, in the 30s, that the idea was that if you observed, I'm going to probably be over at this board the whole time, or no, I'm not. I'm going to walk back and forth and drive you nuts, actually. I know who I am. Not on purpose, I mean, but I will. Okay. So, if you have spatial component, okay, and then running up here can be phenotype, genotype, something in terms of organisms. This spatial component is quite important for Klein theory, okay? And basically what Haldane, and whatever the other guy's name was before him, said was that seeing a Klein in values, some shape, tells you something about the interaction between this and this. And really what they were saying was that the theory would argue that this drop-off or this changeover, whatever the shape happens to be, but in particularly a step Klein, suggests that you have an interaction, because remember this is spatial, you have an interaction between that and that. So a G by E, what we would call a genotype by environment interaction. Okay? So, Klein theory then, in terms of introgressive hybridization, let's just restrict ourselves for the moment in terms of eukaryotes, or talking about eukaryotes. Okay? So, let's say that we're out, and now we're going to get into hybrid zone models in a moment. Oh, by the way, just stop me, raise your hands if you have a question on any of this stuff as I'm yakking. Okay? So, let's say that we're here between two of my plants. We have populations of what look like to be iris fulva, red flowered, about this tall, standing in water. And then we walk through spatial environment somehow, okay, and we end up with something that is lavender or blue flowered, growing in the dry, that we would call taxonomically Iris brevicalis. Okay, this changeover has historically, according to Nick Barton and others, the changeover 80-20, sometimes it's 70-30. So here, you're 80% iris fulva, like, for whatever character you're looking at here. Here, you're 20. Okay? That has been defined as the region of a hybrid zone, where you have highly admixed genotypes. Okay? All right. So, the question is, of course, is why you have this Klein. Nick Barton et al. Said it was not environmental per se. It wasn't that the genotypes were tracking the environment per se in here anyway. The parents would have been in their niches, but the hybrids are always unfit, and so they're there for a different reason. This is their model of hybrid zones. It's still widely applied with no tests of their assumptions. So, and you have to test them. You have to test the assumptions, not the prediction. So one hybrid zone model is called that hybrid zone model I just described to you, or the assumptions of, is called tension zone, or you'll see it sometimes, and actually Nick and Godfrey Hewitt, who really defined this in the 1980s, dynamic equilibrium model. Okay? This is actually, I think, and they talked about this, and also tension zone, but dynamic equilibrium, once again, I think is a little bit more descriptive of what's going on according to their model. Now, let me tell you what their assumptions are. For a hybrid zone to be there, and then we'll move on to other models, for the hybrid zone to remain, regardless, okay, they're assumptions, not observation. Hybrids always less fit. Now they do mention as a throwaway line in their 1985 review, well, maybe some alleles that rarely would get across introgress that were adaptive, but you wouldn't detect them anyway because they would go to fixation and it would look like allele sharing just from the ancestor, which is a good point if they actually did that, but once again, you have geography to test that, okay? But anyway, hybrids, that's one. And so then you have continuous. They call it migration, which is fine, I think. I'll get out of your way in a moment for you that are on my left. Okay, so assumptions are hybrids, and this is where the dynamic equilibrium is. Hybrids always less fit, continuous migration of parentals into zone, into this, to hybridize. Why is that necessary? Because these are all being removed, right? So the only way you get, under their model, the only reason you should see a hybrid zone, the only possibility of seeing a hybrid zone is if you have the parents continuously interacting with one another. If one of these parents goes extinct, this hybrid zone, their expectation, this hybrid zone goes extinct. Right? You don't keep a bunch of hybrids around plus this parent because all of these guys are unfit. Okay? These are assumptions, not predictions. Please, 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 please understand that. Why do I harp on that? I don't know what you're working on. Well, I know some of you are working on Hox genes, okay? And some of you are working on plants and things like that. I know a few of what you're working on. But regardless of what you're working on, you need alternate hypotheses that you're testing. That's part of science. You always need to ask yourself, do my alternative hypotheses give me very different predictions? Because what I'm going to show you is a diametrically opposed model of hybrid zone evolution here that was proposed by somebody else, and yet the assumptions are very different, yet the predictions in algebra and calculus are identical. They overlap one another. So if you tested the predictions, which is what you see, I've got examples in my book where someone in evolution last year published a paper that said this is a tension zone because we see step climbs. They did not test it. They saw step climbs and they said, see, the hybrids are unfit. Okay. But if you test the assumptions, then you can get somewhere with whatever models you're looking at. If your predictions overlap, maybe your models, your hypotheses or alternative actually have really different predictions. So test the predictions. Quite often in science, that's not the case. Yes? We will talk about that in a moment. The question is geographical distance, environmental distance. So this is so Haldane's model was, this is tracking the environment. This is an environmental change. Tension zone, this is an ecotone. But there's a spatial dimension to this. But we'll talk about in a little while why we may not really see step climbs. Why we don't see them in our plants. Why they probably don't see them in many animal systems if they look at detail at it. But let me write up the alternative. Yes. The 80-20? Okay, these are your Y is a measurement of phenotype or genotype. Usually now it's genotype, right? So we're looking at genomic information, not whole genome, but anyway, so what we're saying here is that iris fulva has these alleles, and then as you walk through here, as we walk down a bayou and collect and collect and collect, you change over from 80% genome being fulva to 20%, and it's just the definition of a hybrid zone. We could do it 70-30, we could do whatever, but it had to do originally with, yeah, so that's what it is. It's a changeover from that. But this could be any characteristics. In Haldane's model, it's obviously phenotype, as he's talking about, right? And he used data from deer mice to demonstrate this. Deer mice are this little rodent that we have, Paramiscus, this is a genus, in North America. And he applied it to different colors, coat colors, and they were on different substrates. So a dark substrate has a mouse with a dark back. I mean, this is really one of these classic, beautiful natural selection experiments. And as you go through this ecotone from dark to light sandy color, you end up with transitional zone in those coat colors where you then end up with a light coat color here. So it's tracking the environment. So in 1948, when he published that paper, and later on, those were the characters. So they were quantitative traits. And you can still do that. People actually do that with morphological, like length of legs in crickets, or plumage, bird plumage, male coloration. And they actually look and see where the plumage characteristic, I talk about this in the book, where the plumage characteristic cline is versus others. And they see that the male plumage is way out here. It turns out from experiments and observations, the females are selecting for the odd coloration in those males. Blue females like yellow males. Okay, so yeah, so it could be phenotypic traits, but generally we're talking about genotypic. Okay. So the other model I'll talk to you about, and boy is my handwriting terrible. Bounded, that's a B, bounded hybrid superiority model, hybrid zone model. This one was put together by Bill Moore. So this is Barton Hewitt, etc. And this one, bounded hybrid superiority, you have exactly the same observation out in nature. Bill Moore worked on birds, okay, flickers in North America. And he looked at flickers across the range systematically, but then also in overlap areas in sort of the middle of the U.S., middle of North America. His assumptions were that his major assumption that matters is that hybrids more fit in the ecotone hybrid zone. Okay? I don't know if you remember, but I showed you yesterday some data for the sagebrush and their initial data that they had on survivorship and all that. The hybrids were more fit in the hybrid zone. That was a reflection of falsified the tension zone and supported this. It was consistent with this. So, bounded hybrid superiority, tension zone. In terms of what the hybrids are doing, completely different. Also, this is an environment-dependent model. And when I pointed this out we pointed it out in 95 in a tree article and then 97 in my book. People got a little bit upset, but I pointed out that this was environment independent. It does not, with respect to the hybrids, it does not matter where you put the hybrids here in this geographic space, phenotypic space. They're always 1 minus S if you look at the algebra. So they always have less fitness than the parents, which are always 1. It's just the way the model is set. So, hopefully you remember yesterday, hybrids are not always less fit. whether under natural conditions, experimental conditions, period. They're just not always less fit. If they always were less fit, you and I wouldn't be here. Bottom line. Non-introgressed humans would be the only things here, right? Across the range, as an example. If they were always less fit, you wouldn't see introgression of male plumage coloration in birds. If they were always less fit, the black wolves would not be doing better through sexual selection and survivorship. If they were always less fit, Darwin's finches, the beautiful story we have, wouldn't be real. Okay? So this is demonstrably incorrect. And what we see is every time it's tested, it comes out that hybrid genotypes have a range of fitnesses. Some are lousy. Some have low fitness, like that. Okay, some with zero fitness. Remember yesterday's talk. Some are equivalent in fitness in certain environments to their parents, maybe higher in others, maybe lower in others. And hybrid genotypes, relative to one another, vary in fitness. Just like population genetics tells us about any genotypes. G by E is how evolution works. Okay, yes, genetic drift is important, I'm sure in certain cases, but Darwinian evolution depends on G by E. Okay, so these are two starkly different hybrid zone models. And so applying Klein theory, I'm just telling you, you can do this and you probably can get your papers published, but if you do this and you see this and you conclude that you either have a tension zone or a bounded hybrid superiority zone without testing in any other way, you should be ashamed of yourself. No, I mean, you should worry about it. It's like having phylogenetic discordance and going, well, I'm working on plants, so it must be introgressive hybridization. Or I'm working on animals, and we know it's not important in animals, so it can't be that, so it has to be incomplete lineage sorting. See, you're bringing an assumption in with no test. And you're saying, this is my prediction. I'm going to have phylogenetic discordance because, and you fill in the blank. Okay? So in these cases, if you see this, which almost certainly you probably at some scale will see it in hybrid zones, it does not mean that all hybrids are unfit, and it doesn't mean that hybrids are more fit, all hybrids are more fit or largely fit in that ecotone. In fact, it's going to vary. Yeah. That's correct, which is what you see in hybrid zones. Well, in animals and plants, well, I'm going to show that, I'm going to put a complexity here in a moment. But where we see these kinds of overlaps, okay, not every time, but almost every time, where we see these kinds of overlaps and hybridization is where you've had, especially if you've had secondary contact, the ranges of those are presumably are very often in ecotonal areas. In fact, so in other words, the margins of the niche for both species. Okay? So, but those ecotones move, right? I mean, across the landscape, your environment changes through time due to climatic perturbations. So it's not like those ecotones stay locked in the center of North America when there's an ice sheet there, right? Those ecotones will move off to the side or down south or whatever in North America. Now, let me throw a spanner into the works, as they say in Australia. Not really, but let me give you the real complexity of this situation. So we're looking at this, and that's real. I mean, you see these kinds of step climbs, okay? So in our irises, and I don't have this slide, in our irises when we look, well, that was wonky. In our irises, when we look across the genome, very often, looking at different loci, you'll see this repeatedly. It doesn't go to zero. You know, it's out here somewhere. But multiple step climbs, coincidental step climbs. This was supposedly indicative of You wouldn't get these unless the hybrids are all less fit. Okay? Yes?
How does this talk about, or how does this indicate the fitness? Because they are just showing the phenotypic or genotypic gradient, right?
Yeah.
So it doesn't indicate the fitness.
So, first of all, this is an assumption of the model. This is an assumption of the model, okay? Of both models. Those are assumptions. So just looking at this, we can't really say fit or unfit, except that the prediction is from this model in particular is the steepness of that indicates the strength of selection against the hybrids. That's how people will do it. So if you have that, the argument would be that locus and that locus differ where there's still selection. This is this model. All the hybrids are crap, but they're not as big a crap as that. Sorry for that scientific terminology I just used. They're not selected at that locus. The hybrids are not showing as much negative selection as they do at that locus. So the shallowness there of that, rather than a steep step climb, is indicative of that under this model now, which has that assumption, right? That's how Klein theory tells you something about the distribution. Just in general, it'll tell you something about the distribution of that too, according to Haldane. But Nick and Godfrey use this here, the Klein theory here, to say the steepness of this doesn't really, they're not saying it has anything to do with environment. They're saying, no, it just shows us how bad the hybrids are at that particular locus. Yeah?
But this theory doesn't take into account the fact that there could just not have been enough time for, colonization of that space between the two parent or species or populations.
So you're saying what's here?
So it seems to be built on this ecotone kind of a thing, but that's not necessarily really, because it could just be that there has not been enough time for range extension, and in effect what the hybrids are doing is that they are just occupying some, empty space between them. Not in a geographical sense, but...
No, you're exactly right, and that's what he's arguing. Okay? What Nick and Godfrey are arguing is that this is just artifact of where the parent populations happen to come together. It's part of the environment independent portion of this. And so, yes, it's true. They maybe they're going to do this, the parents, which would not, in fact, be incompatible with a lot of data that people see. And doing this can mean that you just have increased integration, right? And so, yes, absolutely. What I'm trying to tell you, though, guys, is papers are still applying this on its own. 2015, I'm sure there are, especially in birds. They love it in ornithology. I have nothing against birds. They're tasty, okay? So that was rude, wasn't it? But no, bird studies are great. They're lovely. Darwin's finches, ducks, all sorts of things that show reticulate evolution all over the place. But ornithologists, particularly those who are at Yale or one of those kinds of places, just love this stuff because they're assuming that they assume this. They don't test for it. So your point is well taken, but let me give a little bit more detail of what another hybrid zone model that Rick Harrison and Dan Howard and Rick Harrison put together. I don't know if you can see this, but it's the Mosaic hybrid zone model. And I think it is actually much more realistic. And the Mosaic hybrid zone model okay, posits that if you have, and I'm not going to draw the clines right now, but if you have fulva and brevicollis on the landscape, instead of actually going from fulva habitat into brevicollis habitat on a larger scale anyway, you actually have a mosaic of habitat scattered around. So let's say something like that, okay? And so yes, on a larger scale, in this area and in these, especially where they're butting up against and overlapping, if you zoom out, you can get this. This was Dan and Rick's argument, okay? This is their model. I didn't come up with it, although I think their model is very, very good. I will say something. Their model, and Dan and I are very good friends, and I pointed out to him, I'm about to write about this in these books, and I'm going to tell them that your model is environment independent. And he said, no, it's not. And Rick went nuts. And I said, yes, it is. Guess what the hybrids are here? Because these are biological species concept folks. The hybrids are always 1 minus S. Regardless of where they are, they're saying there's no hybrid environment in here. There's no niche for the hybrids. But let's just talk about the spatial outlay here rather than the assumptions. And so with this, if you turn it back on its side and you're walking through, or you walk through like this, and they're scattered, now these are going to be all over the place in here, F's and B's interdigitated. What you predict from that, if you get fine enough scale, is reversals in frequency rather than a smooth climb. Because you're going to walk in between B and F repeatedly as you walk across this. Whoops, sorry, I got a bug friend. This is more reflective, I think, of nature, particularly if you're working on plants, but also animal systems. In fact, they were both zoologists working on insects for the most part. Rick passed away recently, sadly. He was at Cornell and worked on crickets and lots of different things. Did beautiful work. Okay, I cite him in the book about don't invent new terms. He had an evolution paper on that. It was beautifully written. Stereotypically Rick. And so, but this is what you expect if this is correct. But once again, in terms of the estimation of importance of hybrids, they always said, their model said 1 minus S. So no, in this mosaic of environments, this goes back sort of to what we were talking about. In this mosaic, there are no, according to their model, there are no that. Which, of course, we now know is naive based on all the studies that I've told you and what you can read in the book. Okay? It's not always true that all hybrids are unfit. Period. So, but this is what you expect. But if you zoom out, you can imagine that those peaks are in here, right? And this, Rick and Dan pointed this out. If you zoom out spatially and you do your sampling a certain way, you will end up with this. Because they had to explain why people saw this. Because you do. Okay? Ours, I started to draw this earlier. Early on, we did a lot of different markers in single hybrid zones. And it was like that. And they were coincident in their changeover, and it was within 50 meters. Okay? I mean, you talk about looking like really strong selection against hybrids, if that's what was going on, or bounded hybrid superiority, if that's what was going on. But then when you actually mapped this out on the environment, you found out that this changeover was from largely iris fulva to largely brev, but these were almost all hybrids. And these were hybrids, and these were hybrids, and these were hybrids, and then you map it out ecologically. Where are they growing? Is there water? What is the associated vegetation? And it looks like this, except they're all hybrids. And they're scattered. When you look at it, certain hybrids are occurring in certain places. But it gives you this, which belies which one of these it is until you then, which maybe one of these kinds of models it might agree with, until you go in and you do some other tests. You do reciprocal transplants, you do, where are they occurring kind of analyses. And then it tells you something more about how the hybrids are doing or not doing. Yeah?
If you're not hybrids, you would get some loci for which the slope would be much smaller, right?
Well, that was just it. We didn't find that. In this analysis, now we have. But in this initial analysis, the point was when we saw these stepclines and we published it, we said if we had have just applied the tension zone model or the bounded hybrid superiority model, we could have said alternate conclusions without doing anything else. The only thing that told us that these stepclines had something to do with the environment was then breaking it up, making more of a mosaic of it, because we were able, around each one of those plants, because they're long-lived perennials, we could actually tell what the environment was. Not every one could we say, oh, it's there because it's in a wet environment, but certain genotypes we had were. But what I'm saying is, depending on our scale, if we step back too far, because this is 50 meters is where that 80-20 is in this particular hybrid zone. So that was too far back, right? We had to go in and really look where the plants were and what they were consisting of in terms of their environmental associations. Sometimes 200 kilometers isn't too much far back, depending on your organism maybe. That's probably always too far. But anyway, you get my point. Yeah.
I don't know whether the question is finished, but instead of going from iris vulva to the iris other, we do the other way around. The graph will be like this.
Yeah.
The increasing.
Yeah, if you flip, if your phenotype, genotype is, this is the percentage of Brev, yeah, it just flips it. It's a mirror image.
No, but the graph will be like this now. It will increase instead of rise, you will get an increase.
What I'm saying. Yeah, it will be, and you can do that. And in fact, sometimes if you're doing these kinds of analyses, let me encourage one other thing if you can do them. I want to get to your question and would the Kleins shallow out? The other thing about this model, and I think they're correct, is if you come at this not at a 90-degree angle, okay, our hybrid zones often are linear because they're running along, for these two species, actually are hexagona. They're linear because they're tied to linear waterways, okay? Hybrid zones in mammals and birds are often linear because a highway's there. And so you can sample them easier. You're not going to helicopter, well, we helicoptered in Suriname all over the place, but usually you're not going to do that. You're going to follow, it's like fossils, you're going to follow where you can do it easiest, right? So what I'm saying is that if you take a hybrid, let's say that you have a spatial distribution like this, and you're wondering whether or not your zone is really like this. So this is one form and this is another. You're wondering about this? What we do, we did this in fire ants, for example, when we were working on Solenopsis in the southeast. There were invasive forms that came up from South America. Yes, we did that. The student did that. But he also did that and that. Because if you come at it from an angle, you're going to cut these. And you will get, if this is real, okay, if it's a mosaic, or even if it's a tension zone versus that, you're going to change the angle of the slopes and you may shallow them out. And that can tell you something about same loci. It's just that you're sampling across at different angles. You actually can cut that cline at a different angle, and so the peaks are going to change if it's mosaic, and your cline shape can change. And you can use that for inferences if you have other data about environment, etc. In the Solenopsis, for example, we concluded that it was largely a tension zone with all of the available data. So I'm really not only interested in introgressive hybridization, but in those fire ants, just in the pattern and the way that hybrid mess has evolved over time since early 1900s, probably more of a tension zone. Yeah.
If the environment is represented on your x-axis, then the slope shouldn't change. If it's space, it's understandable, but if it's environment that you're modeling on the x-axis, then no matter what direction you're approaching from, the slope of the line should not change.
Exactly, but that's why I'm saying if it's mosaic, it's environment dependent. If it's tension zone, it's not. So if it's like this and you cut and the environment changes, you will see slope changes. Okay? So that's comparing. What we're saying is that when we did this with the fire ants, we wanted to know if the hybridization we saw maybe was environment independent or environment dependent. We could not do, we did some, the student did some, where are the ant beds, but we really couldn't do what we did with the irises, which is go around every individual and ask what their environment was. So instead, people use these analyses quite often. We cut across there to test whether or not, whatever the models, whether or not our slope changes were indicative of environment dependent or independent. The answer is it was independent. We would suggest it was more like tension zone. But yeah, you're right. If there's no correlation with the environment, it's not going to change. That's the point of cutting across it, right, at different angles. If this is so, okay, that's what you have there at a fairly large frequency. So this is what we were looking for. With that, we were really asking if you cut through there, if you would see that, something like that. And that's not what we saw. So it didn't look like it was a mosaic zone between these two. Didn't look like they were homing in on different ecological settings. Yeah. Yes. I'm sorry, I cannot hear you. It's my ears, it's not you. Thank you.
Yeah. So on this fluctuating cline, where will you place these two species, like Babaco and Fulva? I didn't understand that. Can you just tell?
Where are they?
Yeah, on that fluctuating cline,
Uh-huh.
the down.
Uh-huh.
Yeah. Where will you place the Babaco and Fulva? Yeah.
In our case, Brevicollis and Fulva are very different environmental settings, but Brev are in the dry and Fulva is in the wet environment. And so, depending on what you wanted to draw here, whether it's Fulva or Brev, it doesn't really matter, but this would be more Fulva-like. That would, let's say it's Fulva. That would be more Fulva-like. That would be more Fulva-like. That would be, this would be, and then down here in the valleys, you would have Brev. You can flip it around. It doesn't really matter. So, but these reversals, they're called, Dan and Rick said, look, these kinds of reversals, as you're walking across the habitat, the hybrid zone, are indicative of a mosaic hybrid zone where it's environment dependent, rather than a tension zone where you walk across and it's just going to be a smooth climb because it's environment independent.
So where do the hybrids lie in this case?
Well, first of all, in Fulva and Brevicollis, these are the kinds of patterns we were getting until we actually went into the habitat. But where the hybrids lie in here, these are hybrids, okay? But that's why I said more Fulva-like, et cetera. As you walk across the environment, there are Fulva environments with a few Fulva individuals there. But in this particular zone of ours, it's mainly made up almost exclusively of hybrids. But where the hybrids would fall here, if it was you're walking across between two and you basically have that, but now you've blown it up, the hybrids are going to fall in, because this is phenotypic or genotypic information, they're going to fall in between those valleys and peaks. The question is not genotype, so this is genotype, phenotype, just like this, okay? Not where they fall here, to me anyway, the important question is that an environment that actually is a niche that they're less or more fit in or than their parents or whatever? And those are the real key experiments for this that people have done. You know, they're doing them in both plants and animals, but those are the tough experiments.
So it's like they're tracking the environment, can we say from here?
Yeah, absolutely.
Yeah,
If it's the
yeah.
Mosaic zone, they're tracking the
They're
environment.
tracking the environment.
Absolutely. Yeah. But the hybrids may not be. Okay, not all the hybrid genotypes are probably going to are here between the two species because they're fit in that environment, right? They're recombinants, and so some, that's what we detected from this, some are always associated, some kinds of genotypes are associated with certain environments. But some hybrids didn't show any correlation with any environment, they just looked like it was idiosyncratic.
So they would lie somewhere outside the client site, like somewhere the...
No, they're still, remember this is phenotype.
Yeah, that's a phenotype, but as you said, some are not correlated with the habitat per se.
No, what I mean is as you're walking through here, you're hitting different habitats and getting the same genotype in the different habitats. Yeah. So they may be in different, this may be this, if this is what I'm talking about, this is the same genotype. And it's all over the place in terms of habitats, maybe. So
What about the fitness of the hybrids? Does this model talk about anything about that?
the fitness of the hybrids in the Mosaic model, they were always less fit than the parents.
So just like the tension zone.
Exactly like the tension zone. And that's why I pointed out that really the only environment-dependent model was this. And people finally saw that, and they've modified it a little bit, but the genomics is taking care of that anyway, because now we have different ways. I don't mean whole genome sequencing, by the way. I just mean being able to look at a lot of genes or a lot of genic regions. Takes care of it because we can then ask about function and things like that. So, but yeah, but people are still using that. In fact, just to mention, we use this as we do our genomic walks. And some of the ways that we analyze our data, we look at Klein's shape. But we also compare it to a lot of other things to try to estimate fitness, i.e. experimentation, what's around it in the genome, all sorts of different things. Okay, other questions, or does that answer way more than you were asking for? I know Haldane's paper was what, in Nature? It was very short, and I managed to do that. So.
Mike, you said you often found a line which looked like that, a very gently sloping line.
No, you do, now in the genomic information, with our genome, lots more genetic markers, we will occasionally see that.
Occasionally, okay. So is that dependent? So I'm thinking that would be also dependent on how the gradation in the environment changes. For example, if you have vulva in wet and brevicolis in dry, but you also have intermediate habitats where it's exactly intermediate of dry and wet, and there's also intermediate genotypes between vulva and brevicolis, then you would get a curve like that, I suppose. As opposed to when the wet and dry transition is extremely abrupt, in which you're bound to get a step function like that. So coming back to my previous question, I mean, if the x-axis represents the environment, it also depends on how fine the gradation in the environment is, which will determine the steepness of the curve.
If it's environment dependent,
If
yes.
it's environment dependent.
Yes, and yes, you're exactly right. Yeah, and that's what Haldane wrote, right? He talked about the steepness and width. So the other component here we haven't talked about is the width of the hybrid zone, that 80-20. And that has to do with the steepness, obviously, or the degree that it goes down very quickly. But this width is a parameter for hybrid zone analysis, where they say if you have a very narrow hybrid zone, it reflects really, really very strong selection against your hybrids. As I told you, this is 50 meters. It's a really narrow, quote-unquote, hybrid zone. So until you dig into it and ask questions about hybrid fitness, you draw the wrong conclusion if you're applying this. Draw completely incorrect conclusion that all hybrids are unfit and really unfit. So, but remember, what we're contrasting is environment independent in terms of hybrids now. Okay? This is environment dependent for the parents in the sense of this mosaic of habitats, and environment dependent overall. Because this says the parents are fit in their own habitats, and the hybrids, some of the hybrids are fit in theirs. So this one is the environment dependent, and these two are not. Relative to the relative to the hybrids. But yes, you expect this if your environment is sort of changing. But this is, you only expect it if it's environment dependent. Right. And that's what you have to, I would argue that this doesn't prove environment dependence, by the way. Just so you know, this is not indicative of environment dependence alone. This pattern alone doesn't tell you that. This could just be a neutral integration event. You see these, in fact, gosh, I have to pull up the loci right now that we're writing this manuscript up over, or on. I don't know, maybe 40% of them show that kind of a climb. But when you fit it to a model, asking about, you know what the frequency is, the two parents, and you know across this hybrid zone that we're walking what neutral diffusion should look like, they fit a neutral diffusion model. And it's like that. It has nothing to do with, seemingly to do with environment. It's just, they're not going to fixation quickly, and they're not going way up in frequency on the other side of that species boundary quickly, which would indicate adaptive trait integration. Yes?
Yeah. So this bounded hybrid superiority model is it's an environment dependent. So can we say that hybrids themselves track the environment, because environment won't be stable, right? So hybrid zones will keep on moving. So do the hybrids really track the environment? Can we say this from?
Well, the way that we can say that the hybrids would track their environment is there's a few ways, but I mean I don't want to give you the answer that we can do, and then it doesn't make any sense for animals, for example. But you know, in plants, of course, we do reciprocal transplant of kinds of experiments for this or something else. Common garden, if you will. But reciprocal transplants in different environments, we ask who can survive well where. And that gives us an idea of how the hybrids are doing in certain environments. What environments they're in that are not good, and what environments they're in, their fitness is high. And then we can go back into the hybrid zone and ask the question, where are they? Okay, these are plants. Animal groups are doing very similar things in some groups, where tracking their environment, like the Darwin's finches, they track their environment very rapidly with beak shape changes, right? The wolves track their environment very quickly in terms of, it's loosely, I guess I'm going to say environment, because there is a survivorship benefit to being black there, but there's also sexual selection, so that gets a little grayer in terms of tracking your environment. And so, yes, those are the kinds of experiments that we have out there that are powerful in showing us this. But what we can also do with these kinds of just the Klein shape, and then going into the hybrid zone and asking, where are the crickets? That's what they did, the gorillas. Where are they? Well, this species is on sand. This species is on loamy soils. And we could do the same thing with hybrids. So yeah, that's just in terms of tracking their environment. But obviously there's a lot of other kinds of tests we want to do. What loci are important? Why are they there? Are they adaptive? Do they show selective sweeps? All sorts of different things.
So the
Yeah.
short answer is yes, they do track.
Some hybrids will, but some hybrids will not. Keep that in mind. When we say hybrids, oh my, that's it's an enormous, I'm using that terminology, anything that has some material from two different, or maybe even up to three things. Some will, but some won't, because they'll be unfit, right? And that's an assumption that actually can be tested in a lot of different ways. So, yes, Bill Moore was right about his birds, most likely, with some of his hybrid genotypes. Yes?
And when you say neutral model, you mean that the genotype has no effect on the fitness of the...
That's the assumption. Yeah, so what we're talking about here is, when I say neutral, what you're looking at in hybrids, actually you're looking at it in anything, even if you're doing crosses within or different divergent lines, I guess those are considered hybrids too in Drosophila world and maize. But what I mean by that is that because you know the frequency of the alleles, they're not usually fixed. In other words, 100% of this allele in this species, 100% there. So in our analysis, we needed like 75-25 or 80-20 split. I've forgotten frequency difference. But what we're really talking about is that you do a model based on trying to detect segregation distortion. And what that really means is that as you're walking, so we have genomic markers, as we're walking across linkage group 1 with their mapped markers for our hybrid zone study. As we're walking along here, so you have a graph that looks something like this is what your neutral model suggests should be the frequency of those alleles on the other side of this locus, depending on the frequency differences between the two species. And so as you walk along, this locus is right, let's say, is right here. And if you have segregation distortion, and so here's your confidence intervals. If you have segregation distortion at this locus, you might get a value like that. Or you might, if you have segregation distortion, you might have a value like that. What those values tell you is, here you have an excess of heterozygotes, basically. So, and here you have a deficit of heterozygotes. This would suggest at that locus, across that hybrid zone, this would suggest, well, it's underdominance, okay, but it's selection against hybrids. It's actually selection against the het, so it's got both alleles. But anyway, selection against hybrids at that locus. So if it's down here, as you walk across this, you don't see it getting across, then this is segregation distortion with selection against hybrids. This would be selection for or favoring hybrids at that locus, okay? Because it's getting across at a higher frequency than your neutral model outside of the confidence intervals predicts. It's a frequency-based. You can do it Bayesian, you can do it analytically, whatever. There's a lot of different methodologies. But this you anchor around, okay, it's got this certain frequency. We're going to assume that recombination is whatever, and this is what we ought to get. If they're, and a very large proportion of them hug that line. They're in here, right? 40%, I think, of our loci are like that. They do not violate the neutral model. And then the rest of them violate it. And they violate it in this way or this way, right? It's not all of them are adaptive or all of them are maladaptive. So, and this I'll show to you, I think, next session maybe. Mosaic hybrid, Mosaic genome, next session, which is Monday. I'm getting really confused. I have got to stop and go home. Are you Chinese? You Indian? You know, are we in Beijing? Are we in Pune? I love it, but I'm too old for this probably. A little scattered. No, I'm only kidding. So that's what you do. That's what I mean by neutral, is that it's inside here.
But do you not ever then get would you not expect a horizontal line? I mean, instead of a line, would you not just expect a horizontal line for a neutral model across a gradient?
No, because remember, the reason is it's the way you're doing the analysis, the stuff you're feeding into the analysis. The stuff you're feeding into the analysis, the data, start here, right? So they start here. If you see this, you're not going to use those data. No, but I mean, it's a good question, because if this is what Nick Martin and Godfrey Hewitt said, is that if you see that, right, okay, and they said, well, I mean, yeah, vanishingly infrequently, this could be an adaptive introgression event where it was here, and this introgressed, and then it, boom, and went up, right? So horizontal lines could mean that, but they were correct. I mean, if that's what you saw. But if you think about, once again, take humans. We've talked about this earlier. Take us. So if you look at, that's Eurasia, if you couldn't tell, okay? So if you look at us, there is, and we're going across, right? You're going to start out with Neanderthalensis, maybe like that, but you're also going to have Denisovans, actually, and Neanderthalensis is going to go like that at the end. It's a very large mosaic in one sense, but in terms of alleles, if you only looked, this is Africa, actually, I said Eurasia, and then this would be your Eurasian hybrid zone, right? And this would be Neanderthalensis. But if you go into here, right, it's more patchy. So if you look at Tibet and non-altitude, you're going to have in the hypoxia genes, the hypoxia alleles. So do you see why I'm saying spatial matters? But the cool thing is, this would suggest selection against hybrids for many people, particularly those working on animal systems who think in the biological species concept way. But that's not what's happening here. These guys are hybrids, us, and these guys are hybrids, us, but they have different alleles at different loci that makes them hybrids. These are, let's say that's them. These are incredibly fit up here compared to these, and these are fit down here compared to those. Both hybrids. So that step-cline is indicating something, but not what people often think when they actually test it. Yes?
What if the two species which we are considering are not hybridized, and the line which you are getting is just because of environmental selection?
Absolutely. So Haldane was interested when he was looking at this. This was not hybridization he was talking about. It actually turns out it probably is. But let's assume it's not. He's just talking about stepping from one phenotype to another, tracking the environment. And John Endler wrote an entire book about this. And what John said was, no integration for a moment. Okay, no genetic exchange, no nada. He asked the question, could you start here and evolve in two different directions, basically parapatric differentiation or sympatric it's sympatric. And in this case, this is called primary. Don't ask me why. This is called primary intergradation. And when you have instead range expansion out over time, and finally they overlap. These are not clines. This is geographic edges. This is secondary contact, is what we normally call it. That's called secondary intergradation in the literature historically. I mean, I understand this. Intergradation is like interdigitation. Secondary, they, overlap, but I don't understand primary intergradation. Why, it's intergradation anyway, but this is what they meant. So yeah, that could absolutely theoretically not be a hybrid zone. It could just be tracking the environment, and you see that within species, right? Where you actually have elevational gradients. Now, is it in the past due to the kind of things that we see, like in the sticklebacks, where, they're doing it along a gradient, they're diverging, and they're doing this, where you come in with one form and they spread out, but now Dolph's data suggests that they're starting out as an admixture and then sorting themselves out. So, terms of divergence with genetic exchange again. But anyway, yes. I mean, that's, once again, Klein theory was built around the idea that the environment mattered. And you should see those kinds of clines at some shallow or steep or whatever, depending on how your environment was laid out, which is what you were saying earlier. But then they were used, they were taken and then said we can use these in hybrid zones to tell what's going on. Just to tell you, once again, this is what we're doing as we walk across the genome, which is really sort of cool, right? Because you have a geographic, so we have 480 some odd individuals, for example, in our study, and other people are doing it, across a hybrid zone for 200 and some odd loci, right? And so not only do we have a geographic component, but this is also linkage group 1. And I put this in this book. So the genomic across this linkage group here, you may have a set of things that look like that, like that, like that, depending on which locus you're in. So not only do you have a geographic, but you also have a genomic spatial component that you can look at and ask questions about what's getting across and what's not. So we're using Klein theory. I'm trying to say that Klein theory can be very powerful. I don't want you to get the feeling, oh, I shouldn't. People use it. It's just what you conclude from it. You have to be careful. You have to understand the assumptions and the other models, I would argue. Okay, other questions? We still have lots of time. I'll remind you, Chapter 1, why are we here? Chapter 2, species concepts, and then Chapter 3, testing stuff. Yes?
Can you explain fitness bottleneck?
Fitness bottleneck.
Where hybrids are actually experimented
I'm not sure I don't know exactly what you're asking about, but you're talking about when hybridization occurs, you can have fitness bottlenecks. Is that what you mean?
what happens after that?
So the bottleneck that we're talking about now has nothing to do, that I'm referring to there, is not what we normally think of as a bottleneck. And this is something I actually want to talk to you about in general anyway, so I'm glad you brought it up. But we're not talking about genetic drift, right? Because we're talking about fitness. So, excuse me, I keep stealing the pens. So, when we have, if we look at fitness, okay, I know I keep drawing these kinds of graphs. So if we're actually able to measure fitness of genotypes, so this is fitness, okay, and you can do this without having common gardens in various ways. But if we measure fitness across a group of hybrids, we do expect some to have that kind if these are different genotypes, okay? So it's just a set of hybrids, right? And so when we do experimental crosses with, now we're just talking about eukaryotes, but actually prokaryotes and viruses fit this model too. When you make recombinants, you have an array of fitness results where, once again, let's talk about now phenotypic distributions. So this is a hybrid zone in viruses. So you have initially, you have that, and you have that in terms of the two forms that are hybridizing. They hybridize. Okay? Early stages, it really is more leptokurtic, but let's just keep this for simplicity. Let's say, and those are the two parents still. Lots of hybrids. Then what happens, what you have here is reduced, significantly reduced linkage disequilibrium. Okay? Significantly reduced linkage disequilibrium relative to those, right? Very high linkage disequilibrium in the parents relative to these admixed forms. And what that reflects, of course, is that linkage disequilibrium is being reduced because you have recombination.